Good evening, everyone. Today we have gathered once again to have a discussion on cell signaling. And in today's lecture, I have my colleague, my speaker for today's lecture, Dr. Ritabrata Bhattacharya from PMS College, Malbajar. And also I welcome him. And I also welcome my colleague from the Botany Department, HOD Botany Department, Gurukhan Government College, Mr. Prashant Chetri. Welcome, sir, in today's venture. We are very proud to have you as a special guest for today's lecture. And I also welcome Dr. Ritesh Vishwa, HOD, Gurukhan Government College, and the convener of this lecture series, which we have been organizing, and already we have got great, response from all over the colleges around us. And I also heartily welcome all our students, not only from Gurukhan Government College Zoology Department, Botany Department, and also students from all over the Kalimpong and a major portion of uh, Jalpaiguri district. It's really fascinating to see all these, students and teachers collaborating in this online platform. With no further delay, I would like to request HOD, Gurugathan Government College Department of Zoology, to formally, start the program by addressing today's speaker and our special guest. Over to you,
Thank
Dr.
you,
Ritesh Vishwa.
Dr. Talibdar, my beloved colleague and the main brainchild of this program. And because of his innovative idea, we are here together, sitting in front of our monitor and doing this beautiful amalgamation of ideas, sharing of ideas. Okay, on behalf of the Department of Zoology and on behalf of the Gururathan Government College, I, Dr. Ritesh Biswa, Head and the Assistant Professor of Zoology from Department of Zoology, would like to invite and welcome you all to today's lecture series. Today we are going to listen to Dr. Ritabrata Banerjee once again. Yesterday I already introduced him to you people. Okay, he is a person of merit and he has done a lot of research. He is presently working as assistant professor in the Department of Zoology at Parimal Mitra Smriti Mahavidyalaya. So today he will be discussing on the cell signaling, my dear, and we have the pleasure of presence of today as a special guest, Mr. Prashant Kumar Chetri. He is my colleague and head of the Department of Botany. Welcome, sir, to today's lecture series. And my dear students from our department, from the Department of Botany and from the Department of Zoology of Parimal Mitra Smriti Mahavidyalaya and Kalimpong College, I welcome you all dear students to our lecture series. And I request all of you to stay and utilize this lecture till the end and take as much as you can from this lecture. Ritabrata sir, I welcome you again, and floor is yours. Please go ahead.
Yes, sir. Thank you, sir. Thank you, sir. It is a great pleasure for me. Yesterday already we had a talk, and that was a nice experience that we have gathered. And it is a great opportunity for us also to accumulate all the students from different colleges of North Bengal. And it is a great initiative done by the Department of Zoology, Gurugathan College. And really, it is a pleasure to me being a part of such a monsoon lecture series. So now I am going to talk about the cell signaling. So may I share the screen now?
Sure, sir. Sure, sir. Please do.
Okay. Just confirm me, is it visible or not? That's it.
Not yet, sir. Not yet. Okay, it's coming up now.
Okay, so yesterday there was a problem with the PowerPoint presentation. So that's why I made it in a PDF. So I think there will be no issue. So now we are going to start about the cell signaling. In the zoology major syllabus of the third semester student, mainly we have to focus on two pathways. One is G protein mediated cell signaling via glucagon, And another one is receptor tyrosine kinase mediated signaling via insulin. So now we are starting from the classification of hormone. Basically, we know about the insulin and glucagon. These are the two hormones which used to secrete from the islets of Langerhans area of the pancreas. So in the islets of Langerhans, there are the different types of cells. Among them, from the beta cell, insulin used to come, and from the alpha cell, glucagon used to come. So what is the role of insulin? The main role of insulin is to regulate the blood sugar level, to convert the blood sugar or glucose to the glycogen in the liver or in the muscle. And what is the totally antagonist role of the glucagon, which leads to the breakdown of the glycogen as per the requirement of the blood. Glycogen used to break down by the glucagon, and it used to mix with the blood from the liver and muscle. So now how this glucagon and insulin works in the liver cell or muscle cell, that is we have to learn. First of all, I show you that depending on the chemical nature, there are the different types of hormone. Among them, insulin and glucagon, these two hormones are basically proteinaceous hormone or polypeptides. Depending on the mechanism of action, they involve the secondary messenger or second messenger. So what is that? We know that steroid hormone are lipophilic in nature, so they can directly cross the plasma membrane and enter in the cytoplasm. And finally, directly cytoplasmic receptor and hormone complex can enter to the nucleus and attached on the DNA at a particular area known as hormone responsive element. Over that, they will regulate the transcription of a particular gene, which is required for the purpose of that particular hormone. But in our syllabus, these both hormones are proteinaceous, so they are not allowed to cross the plasma membrane barrier. So they have to sit over the receptor, which is situated over the plasma membrane. So these two hormones, insulin and glucagon, they have the different receptor. First of all, we have to know that generally that glucagon utilize that second messenger molecule cyclic AMP. And another thing, insulin, they use kinase or phosphatase cascade. So these are the two types of hormone. So we have to know how their signaling is going on. So basically we have to focus only to the signaling. That entire part I am just informing you. So now we have to know the mechanism of the hormone signaling. So here you can see that there are the different pathway of hormone signaling. G protein coupled receptor, ligand gated ion channel, enzyme linked receptor, as well as intracellular receptor. That is what I have just told you. That is a steroid hormone. In this case, yellow color, that hormone can enter through the plasma membrane and directly can transport it to the nucleus. But in case of other hormone, they have to they are not allowed to pass the plasma membrane, and they have to sit over the receptor and produce the second messenger molecule. So now we have to focus only two topics. One is G protein coupled receptor, this one, and another one, that receptor tyrosine kinase. That means for the insulin, we have to know about this enzyme linked receptor, that is receptor tyrosine kinase. This is the basic structure of the G protein coupled receptor. Mainly, the G protein coupled receptor, I have written there that G protein coupled receptor bind a ligand and activate a membrane protein called G protein. Basically, G protein is used to contain the three subunit alpha, beta, and gamma. Among them, alpha unit is mainly functional part of the G protein coupled receptor. Initially, alpha unit used to associated with GDP molecule. At the time of activation, they have to bind with the GTP molecule. Then they will be shifted from one place to the another place for the signaling purpose. All G protein linked receptor have 7 transmembrane domain, but each receptor has its own specific extracellular domain and G protein binding site. So G protein binding site you can see here in the cytoplasmic domain, which is situated inner side of the cell, and external surface containing 7 external domains. Now we can see how these things are going on. Glucagon is a peptide hormone produced by the alpha cells of the pancreas. It raises the concentration of glucose and fatty acid in the bloodstream and is considered to be the main catabolic hormone of the body. So basically, what is the purpose of the glucagon? The purpose is to produce the glucose. That glucose is stored in the muscle or liver cell in the form of glycogen. So what we have to do? We have to break down the glycogen and transform it into glucose. Then it will be mixed with the blood to fulfill our blood sugar level. The pancreas releases glucagon when the amount of glucose in the bloodstream is too low. Glucagon causes the liver to engage glycogenolysis. That means breaking down of the glycogen molecule into the glucose, converting stored glycogen into glucose, which is released into bloodstream. As a result, blood sugar level will be enhanced. So now, how that signal used to do it? Here you can see a diagram. This is a receptor. This receptor is G protein coupled receptor. From the outside, glucagon will come and sit over the receptor. So below the receptor, you can see here alpha, beta, and gamma subunit. Alpha, beta, and gamma subunits are there. Alpha remains associated with GDP. So whenever glucagon will come and sit over the receptor, then GDP is converted into GTP. So as a result, alpha subunit will be released from the receptor and move another side. So as a result, after binding, you can see this diagram. What is happening here? Here you can clearly see like that receptor, after attaching with the glucagon, alpha unit already released from it and it is going to the left side. As a result, what will happen? That alpha GTP unit or subunit that will go and activate another enzyme that is known as adenylate cyclase. So what is the purpose of this enzyme? The main purpose of this enzyme to convert ATP to cyclic AMP. So basically, the activation of the G protein, that means the release of alpha subunit with the help of GTP and activation of the another enzyme adenylate cyclase for the conversion of ATP to cyclic AMP. Now you can clearly see ATP is converted into cyclic AMP. What will happen then? That cyclic AMP will activate protein kinase A. That protein kinase A is another second messenger molecule that will be activated with the help of cyclic AMP. After activation of protein kinase A, what it will do? It will further activate another enzyme called phosphorylase kinase through the phosphorylation. You can see here PPK associated with phosphate group. That means it's already phosphorylated. So another molecule gets activated. That is a cascade of reaction which is happening through the activation. And that activation is mainly done by phosphorylation. So after activation of the PPK, what is happening? Another enzyme that is written here as PYGB, that means glycogen phosphorylase B, that remains inactive. With the help of PPK, that becomes activated and becoming glycogen phosphorylase A, that is the active molecule. And this is the final molecule which help in the synthesis of different enzyme for breaking down of the glycogen molecule, that is known as glycogenolysis. So for doing this thing, glycogenolysis, which is a part of carbohydrate metabolism, that signaling cascade should be followed. So I am just going through those points. Glucagon binds to the glucagon receptor, a G protein coupled receptor located in the plasma membrane of the cell, which is shown here. The conformation change in the receptor activates G protein, a heteromeric protein with alpha, beta, and gamma subunits. When the G protein interacts with receptor, it undergoes the conformational change that result in the replacement of GDP molecule that was bound to the alpha subunit with a GTP molecule. So GDP will be replaced by GTP. As a result, alpha subunit becomes activated and it will be released from that coupling area. And as a result, GTP will go and activate another enzyme called adenylate cyclase. The alpha subunit specifically activates the next enzyme in the cascade. That enzyme is adenylate cyclase. Adenylate cyclase converts ATP to cyclic AMP. And then finally, cyclic AMP will activate protein kinase A, which is shown here in the green circle protein kinase A. Then this protein kinase A, after activation, it will activate another enzyme, phosphorylase kinase, that is PPK. After that, that activated phosphorylase kinase will activate PYGB into PYGA. That means it will form glycogen phosphorylase in active state, and which will undergo the metabolic activity by producing the enzyme. Those enzymes are required for glycogen breaking down or glycogenolysis. So that is the entire process of the G protein signaling. Now here, that is a schematic thing you can understand much more easy way, that you can see here glucagon is binding on the receptor. Receptor is shown here in the green color, and receptor remains associated with alpha, gamma, and beta subunits. After binding, what is happening? Alpha subunit is released and going to the adenylate cyclase along with the GTP. As a result, adenylate cyclase gets activated and it will convert ATP to cyclic AMP. Cyclic AMP will activate protein kinase A. After that, protein kinase A will activate phosphorylase kinase. Then phosphorylase kinase will activate glycogen phosphorylase. After that, glycogen will start to break down with the help of enzymes, which will be synthesized by the glycogen phosphorylase. So this is the entire process of G protein mediated cell signaling via glucagon. Now we are coming to the next topic, that is signaling by receptor tyrosine kinase via insulin. So I told you that the role of insulin, that is just antagonist to the glucagon. What they will do, insulin used to store the glucose from the blood to the liver or muscle cell in the form of glycogen. So what is the basic fundamental things of the enzyme linked receptor? You can see here two diagrams. What is happening here? They have the three domains. One is external surface or outer side of the cytoplasm. Another portion is the transmembrane domain. And finally, at the cytoplasm, there is a cytoplasmic domain. So whenever the ligand or hormone will bind to the extracellular surface, that time different enzymes will be activated in the cytoplasmic domain. So what is written here, that enzyme linked receptor are cell surface receptor with intracellular domain that are associated with an enzyme. In some cases, the intracellular domain of the receptor itself is an enzyme. That's why we are telling the term tyrosine kinase. So kinase means phosphorylating enzyme. Other enzyme linked receptor have small intracellular domain and interact directly with an enzyme. When ligand binds to extracellular domain, a signal is transferred through the membrane and activating the enzyme. Activation of the enzyme sets off a chain of events, that means cascade of reaction. That is the same thing which is happening in the case of glucagon. That is the same thing will happen in case of insulin also. Just you follow that. Here, that I told you from the beta cell insulin used to come. So what is written here, insulin is the peptide hormone produced by the beta cells of pancreatic islets. It is the main anabolic hormone of the body. Why it is anabolic? Because it is responsible for storage of glucose in the form of glycogen. Secondly, it regulates the metabolism of carbohydrate, fat, protein by promoting the absorption of the glucose from the blood into the cell of the liver, fat and skeletal muscle. The insulin transduction pathway is a biochemical pathway by which insulin increases the uptake of glucose into fat or muscle cell and reduce the synthesis of glucose in the liver. And hence, it is involved in maintaining glucose homeostasis. So that means blood sugar level will be maintained by the activity of the enzyme, insulin. So here you can see that diagram, you just follow the diagram. After insulin enters in the bloodstream, it binds to the membrane spanning receptor tyrosine kinase, or that is simply receptor tyrosine kinase. Here you can see that insulin receptors are there. Insulin is coming through the blood and it will sit on the receptor at the extracellular domain. What is happening? This glycoprotein is embedded in the cellular membrane and has an extracellular receptor domain made up of two alpha subunit and an intracellular catalytic domain made up of two beta subunit. What is alpha and beta? Basically, I can show you one diagram. Here, I am enlarging that. Now, is it visible to you? You can see this diagram. So here the outer side of the plasma membrane, that is the alpha unit. Then inside there is a beta unit of the receptor. So just focus on the alpha unit, which is responsible for capturing the hormone, and beta unit is responsible for the enzyme activity which will happen for cascade of reaction within the cytoplasm. So initially what is happening, whenever that insulin will come, you follow that second diagram, that blue color insulin. So whenever in the outside insulin molecule will come, you can see that beta subunit gets phosphorylated. Now is it, I think it is visible. So now you can see that beta unit gets phosphorylated. In the third step, you can see I have written there activated insulin receptor protein kinase. So activated insulin receptor protein kinase, that is the enzyme which is associated with the beta subunit. Then parallelly it will activate another substance that is called IRS1. So what is IRS1? We will come to know later. So what is happening? First, insulin will sit over the alpha unit. It will activate the beta unit through phosphorylation. As a result, insulin receptor protein kinase gets activated, which will further activate IRS1 molecule. In the next diagram, you can see IRS gets phosphorylated, hugely phosphorylated. So this phosphorylation is done by insulin receptor protein kinase. Initially, you can see here IRS1 without any phosphorylation. But in this step, you can see IRS1 with 4 phosphate molecule. So naturally, that huge phosphorylation happen with the help of insulin receptor protein kinase. So that enzyme will phosphorylate IRS molecule. Now, if we come to the next diagram, what will happen? That IRS molecule will transport that phosphate group through a channel to the plasma membrane. That phosphate group used to transmit it or transport it to the plasma membrane through a channel. So what is the channel? That, oh, sorry. So you can see here that channel is called PI3K. That means PI3 kinase. So PI3 kinase will transport that phosphate group from the IRS molecule or IRS1 molecule to the cell membrane. In the cell membrane, there is the existence of another molecule known as phosphoinositol phosphatidine. That is PIP2. That PIP2 will receive another phosphate and becoming highly phosphorylated PIP3. So PIP2 and PIP3, they are existing in the plasma membrane, not in the cytoplasm. What is happening after activation of the receptor tyrosine kinase? That means the beta unit of the receptor, then it will phosphorylate that IRS molecule. IRS will transport that phosphate group to the PIP2 via PI3 kinase. As a result, PIP2 will be converted into PIP3. And what is the role of PIP3? Then PIP3 will activate another enzyme called PDPK1. So PDPK1 is the enzyme which is situated in the cytoplasm. So that will be activated by PIP3. And finally, PDPK1 is responsible for activation of AKT, that is the main second messenger for the signal transduction. And AKT will undergo different activities. So now we are going back to that initial slide. So here also you can follow the diagram. After insulin enters in the bloodstream, it binds to the membrane spanning receptor tyrosine kinase. This glycoprotein is embedded in the cellular membrane. Mainly the thing is alpha subunit, they will receive that blue color or sky blue color, that is the alpha subunit, and violet color is the beta subunit. So we come to the end of the beta subunit, you can see here IRS molecule. So what is the IRS? IRS means insulin receptor substrate 1. That is the insulin receptor substrate 1. So what will happen? One of the phosphorylated tyrosine residues of the insulin receptor attracts a protein called IRS1. After binding, IRS itself phosphorylated by insulin receptor protein kinase. So I told you that insulin receptor protein kinase vigorously phosphorylate that IRS molecule. Now, what's the role of IRS molecule? IRS molecule will transport that phosphate group to the plasma membrane. IRS1 molecules are called adapter protein because they themselves do not activate anything. Phosphorylated IRS1 acts as attachment point for lipid kinase called phosphoinositide 3K or PI3 kinase. So what they will do, IRS will transport phosphate group through PI3 kinase. So PI3 phosphorylates PIP2, which I shown you. PIP2 is situated on the plasma membrane. So PIP2 will be activated. PIP2 means phosphatidyl inositol 4,5 bisphosphate. That will be converted into PIP3. So you can see here, it was initially 4,5 bisphosphate. After receiving another phosphate group, it became phosphatidyl inositol 3,4,5 triphosphate. So from where they received that phosphate group? They received the phosphate group from the PI3 kinase molecule. That PI3 kinase will transport that phosphate group from the IRS molecule. Then after receiving, they become PIP3. What is written here? It travels along the plasma membrane to activate PIP3 dependent protein kinase 1, that is PDPK1. PIP3 dependent protein kinase 1, that is PDPK1. That is again. So PDPK then activates another protein kinase B or AKT. So AKT, our main target, that is the main second messenger. It is not membrane-bound protein and able to diffuse around the entire cell cytoplasm. What is the role of AKT? AKT1 stimulates the movement of glucose membrane transporter or GLUT4 to the cell membrane for the reabsorption of PI3 kinase. Then after PI3, in the diagram it is not drawn, but in my hand. So after activation of, they will activate the AKT. Main purpose of the AKT to shift that GLUT4 receptor to the plasma membrane. Without that particular channel, receiving of the glucose is not possible. So now you can see here that GLUT4 will go to the plasma membrane and they one also deactivates GSK3 glycogen synthetase kinase 3. You can see here AKT deactivating glycogen synthetase kinase 3. So glycogen synthetase kinase 3, that means it will help. Generally, the purpose of the insulin is to receive the glucose molecule to the liver. So glycogen synthetase kinase are very essential for glycogen synthesis. So basically, after receiving the glucose, that will induce that glycogen synthesis. So basically, glycogen synthesis will be inhibited as a result smoothly on. After that is the main target of signaling cascade of the insulin. Now this is the diagram. Again, I am showing you. I think you just follow the diagram that receptors are situated. Here you can see outside and the plasma membrane. That is the receptor without the insulin. Next diagram you can see after receiving insulin, then acted with the help of phosphate group. That means That is insulin receptor kinase activation. Next diagram you can see, they will bring that IRS molecule near to the beta subunit. After coming the IRS molecule, that will be phosphorylated. In the next diagram you can see, IRS molecule is vigorously phosphorylated. So after phosphorylation, that IRS molecule will act transport that phosphate groups to the plasma for phosphorylation of PIP2. So that is the transfer molecule that is called phosphoinositide 3 kinase that will transport phosphate group to the plasma membrane, and PIP2 will be converted into dependent kinase or PDPK. That PDPK is responsible for activation of AKT molecule, which will act as the main transcription factor. That is activation of AKT1 molecule, which will act for the further processes, or it will help to recruit the GLUT4 ion channels at the plasma membrane for receiving the glucose. Parallelly, AKT will inhibit GSK3, that is glycogen synthase kinase 3, which used to inhibit the glycogen synthesis process. So if you inhibit that GSK3, then again glycogen synthesis will going on with the presence of AKT molecule. So up to this is the total receptor tyrosine kinase via insulin. I am audible, na? Is it clear to you? Dear student, is it clear?
Yes, sir. Yes, sir. Yes, sir.
Okay, so I can show you one video. Hopefully that will much more clear your vision.
G-protein coupled receptors, or GPCRs, constitute the largest family of receptors, with more than 800 genes encoding.
Is it visible now?
Yes, sir.
Okay, just follow that.
G-protein coupled receptors, or GPCRs, constitute the largest family of receptors, with more than 800 genes encoding GPCRs found in the human genome. This large family of receptors are very important pharmacologically, as they constitute about 40% of all the drug targets in our cells. GPCRs all have a similar structure, with 7 transmembrane helices embedded in the plasma membrane, with an extracellular N-terminus and 3 extracellular loops, and an intracellular C-terminus with 3 intracellular loops. Endogenous ligands and drugs bind to the receptors either on the extracellular N-terminus and loops, or, if the ligand is hydrophobic, its binding sites may be in the transmembrane helices. Most GPCRs couple to heterotrimeric G proteins composed of an alpha, beta, and gamma subunit. This G protein binds to the inner surface of the receptor and is inactive when GDP is bound to the alpha subunit. This is the basal or inactive state of the receptor in G protein. In the example we are showing here, the GPCR is a beta adrenergic receptor, and the G protein is GS. When an agonist, in this case adrenaline, binds to the GPCR, the receptor changes its conformation in such a way as to influence its interaction with the G protein. This causes the G protein to change its conformation, opening up the guanine nucleotide binding pocket on the alpha subunit and releasing GDP into the cytoplasm. GTP, which is much more abundant in the cytoplasm, will then bind to the G protein alpha subunit. This exchange of guanine nucleotides activates the G protein alpha subunit, causing it to dissociate from the receptor and its beta gamma subunits. The activated G alpha GTP then binds to its target protein to change the amount of a second messenger inside the cell. In the case of GS, its targets are adenylyl cyclases. These are large proteins with two groups of 6 transmembrane segments, an intracellular loop connecting these two groups, and a large intracellular C-terminus. Together, the intracellular loop and the C-terminus make up the catalytic domains of adenylyl cyclase. Without GS-alpha, the two parts of the enzyme rarely come together, and so the level of second messenger cyclic AMP is low. When the GS-alpha is activated, it binds to both the catalytic domains and stabilizes their interaction, making an active enzyme that hydrolyzes lots of ATP to cyclic AMP in the cytoplasm. The increase in cyclic AMP inside the cell stimulates a cyclic AMP-activated kinase enzyme, protein kinase A, or pKa. pKa is a tetrameric enzyme composed of 2 regulatory subunits and 2 catalytic subunits. When 2 cyclic AMP molecules bind to each of the regulatory subunits, they dissociate from the catalytic subunits that are then able to phosphorylate substrate molecules in the cell. In the heart pacemaker cells, L-type calcium channels are regulated by pKa.
So up to this, is it clear? G protein coupled signaling pathway.
Yes, sir.
So okay. So I am sharing the another one. That is a receptor tyrosine kinase. So that will help you to clear your knowledge.
Hello and welcome to today's talk about the receptor tyrosine kinases. I will explain what these RTKs are and explain how they function. Receptor tyrosine kinases are transmembrane proteins which consist of a cytosolic part, and this one has tyrosine kinase activity. Receptor tyrosine kinases are involved in multiple signaling pathways in the cell. They phosphorylate target proteins, which then induce intracellular signal transduction. This can then, in a downstream cascade, lead to, for example, gene regulation. Here we have a monomer of our receptor tyrosine kinase. There is a middle part which is embedded in the plasma membrane of the cell. The kinase also has an extracellular part here, and there is the intracellular part, which contains the tyrosine. The extracellular part over here is for the ligand to bind. So a growth factor, for example, can bind there, and this is called the ligand binding site. Inside the cell, facing in the cytosol, there is the part with the kinase activity, containing our tyrosines. So what are kinases? A kinase catalyzes the transfer of phosphate groups. So look at the kinase in blue. It can take away a phosphate from the trinucleotide and transfers that one to another protein. It can phosphorylate a different protein. This protein, now phosphorylated, can then have downstream functions in the cell. So remember, A kinase is in general something which catalyzes the transfer of a phosphate group. In this case, we have a tyrosine kinase. Most receptor tyrosine kinases are available as monomers first. And in this state, the tyrosine residues are unphosphorylated. Now, once the ligand binds to the receptor site, and this occurs for both monomers, a process called dimerization is taking place. So both monomers change their conformation and a dimer is formed. And this is important because one receptor is phosphorylating the other one. After dimer formation, our tyrosine kinase is activated. And what happens here? We call this transphosphorylation, where one of these kinases phosphorylates the tyrosine of the other one here. After a while, all the tyrosine residues are phosphorylated. So now remember what our kinase is doing. They can phosphorylate different proteins. So we have a protein here in green, and this one can bind to the phosphate residue on the tyrosine. These proteins who are capable of binding to the phosphate have a SH2 domain. Another protein, illustrated in red, binds to the phosphate on the tyrosine here. The receptor tyrosine kinases are able to phosphorylate many different proteins. After a while, lots of different proteins can get phosphorylated by that. For illustration purposes, here we have the green protein, which is now in a phosphorylated state. This can be the start of several different signaling pathways. This protein in green, for example, can phosphorylate another protein downstream, and this in the end could, for example, lead to gene regulations. However, there are plenty of different intracellular signaling pathways which
So up to this, it is clear, na? Because another note is there in the video. That is, other things are there in the video. So that is not essential for you. So only up to this, the activation and the cascade of G protein coupled receptor as well as receptor tyrosine kinase. So that thing, I think it is almost clear. If you have any doubt, you can ask me the questions. Hello, students. If you have any doubt, you can ask me the questions. Am I audible?
Dear
Yes,
students,
sir, you are
please
audible.
respond.
So if you
Yeah,
have
please respond to him. Third sem students?
Hello, Sandipan. Sandipan, am I audible?
Yes. Yeah, you are audible.
আমি প্রশান্ত বলছি এখন স্টুডেন্টরা জিজ্ঞেস
হ্যাঁ,
করবে হ্যাঁ good afternoon everyone. Basically good evening I think now. It is almost 1 hour, no? This lecture, very informative lecture given by Dr. Ritabhatro Banerjee sir, head department of zoology, and it was a very informative and detailed insight into G protein mediated cell signaling via glucagon and receptor tyrosine kinase mediated cell signaling via insulin. Sir, I have one question regarding the secondary messenger. Am I audible?
Yes, sir. Yes, sir. Tell me.
Secondary messenger, in case of G protein coupled this receptor, I think cyclic AMP is the secondary messenger, isn't it?
Yes.
Is there any secondary messenger in this receptor tyrosine kinase mediated cell signaling?
Actually, there are the very much chances of cyclic AMP formation as well as calcium ion also act as the second messenger molecule. Lots of downstream molecule like MAP kinase molecule, AKT molecule, all are the downstream molecule. It depends upon the gene which you are going to activate. So depending on different types of factors, different transcription factor used to be produced as the second messenger molecule. For example, in molecular biology work, we have to, whenever we have to track particular gene level, so what we have to do, we used to do the Western blot of those second messenger molecules. If we are seeing that second messenger molecules are highly activated, that indicates they are responsive genes should be more. So as a result, if you run the PCR of that particular gene, you will get much more higher because it's activator. That means second messenger or transcription factor is higher level in Western blot result. So different types of second messenger can be produced. It depends on the tracking of the gene.
Yeah. Thank you, sir. Thank you. And in case of this, I think there are three types of cell signaling, isn't it? Autocrine, paracrine, and endocrine, isn't it?
Yes.
This insulin comes under endocrine signaling.
Yes.
And glucagon?
Yes,
Both.
it is also endocrine.
Endocrine. Okay. Thank you, sir. Thank you. Sandipan?
Yes, Prashant sir.
এখন স্টুডেন্টরা কিছু বললে আমার কিছু বলা আছে আরো
Yes, you'll be addressing the speaker, obviously. But let us give the students a chance to ask a few questions if they are willing to.
Yeah. Okay. Thank you.
Dear students, you can use your hand tool to let me know who is going to ask the question. Is there anyone? I cannot see any hand tools. I think it is a fantastic lecture.
And Sandipan sir, I can share this PDF with you.
Yes.
So
Yes. Today and today and yesterday
also. Okay. I will convert that in PDF and I will share with you.
Sure, sir. Yesterday, due to electricity failure, I could not join. But today it was such a fantastic lecture, and I enjoyed every bit of it. Anyway, students, any questions? It is a very pertinent topic. You are going to face many questions from this topic in your future interviews and also in NET and any other competitive exams. So if you have any query, you can ask, or otherwise we'll move on with the program. 5, 4, 3, 2, 1. Okay, so I don't think they have any specific questions, but I do really hope that they have learned at least something from this. And I again congratulate Ritobrotoda for such fantastic lecture. And I also would like to share that when myself and Riteshda came out with this, innovative idea of doing such thing, Ritobrotoda was the first, speaker to give his words that you just do it and I'll be there and I'll be taking class on this. So today I actually congratulate him for such fantastic lecture. At the same time, I also take this stage to show my gratitude to him, as from we have had long discussions regarding this program, and he has given me certain advices. And I think the Department of Collaboration between Department of Zoology, Gorgathan Government College, and Department of Zoology, PMS College is really running strong, and in future also will do many such collaborative, initiatives which are really going to help our students. With this note, I would now like to request my colleague, Mr. Prashant Kumar Chetri, a special guest for today's program, to share his views on today's lecture. And now I request Prashant sir. Sir, please.
Yeah, thank you, Sandipan. Thank you for giving this opportunity to become a special guest in this wonderful lecture session. As you know, this is a Monsoon lecture session organized by Department of Zoology. Really, I'm very proud that our Zoology department has organized such a lecture series for the benefit of the student, and I'm sure that today the students of different colleges have benefited a lot from this lecture. And I would like to congratulate Dr. Ritobroto Banerjee for his such detailed insight into the cell signaling mechanism relating to insulin and glucagon. I hope the students have gained a lot of knowledge about this concept concept that is a very important relating to cell biology, where we know that cell responds to different kinds of environmental signals and different mechanism, physiological functions, keeping homeostasis and in the cellular tissue and systematic levels. And I would also like to thank and express my gratitude to the head of the Department of Zoology, Dr. Ritesh Biswa sir, for giving me this opportunity to become a special guest in this online lecture series. And I would like to thank all the students of all the colleges who have actively participated in this lecture series, who has listened carefully. I hope they have listened carefully to the important topics relating to insulin and glucagon, its mechanism, its cell signaling mechanism, and I hope that our department, botany department, will also follow the path of zoology and organize such lecture series in the future. And the zoology department obviously will guide us relating to this kind of lecture series. And really, I'm very proud of Zoology Department, Dr. Sandipan sir, Ritesh Biswa sir, who has taken this initiative to organize such innovative lecture series for the benefit of the students. And really, I'm very, very proud to be a special guest in this lecture series. And once again, I would like to congratulate Dr. Dipavatra Banerjee sir for giving his valuable time, and not only from the presentation but from the videos also. He tried to explain the cell signaling mechanism in mechanism to the student. So I'm very, very proud that such a detailed insight he has given to the student. And once again, I would like to thank all those who have participated in this lecture, to all my dear students from botany department also, who has participated and listened to the lecture carefully. So thank you, everyone. And now I would like to hand over the mic to Sandipan. Sandipan?
Yes, thank you, Prashant sir. You have been a friend, you have been a senior, and you have been a very, very supportive colleague. And I do really hope that our college will keep on organizing these things, and we will definitely do. We are in a strong path, and our collaboration with all the other departments of science in Gurugathan Government College is very strong and effective. And it's only because of that we have been able to take certain these kind of ventures. And we do hope that this kind of collaborative actions will keep on, keep on progressing. This kind of collaborative actions will keep on progressing. Now I request HOD Department of Zoology, Gurugathan Government College, Dr. Ritesh Vishwa, to formally give a vote of thanks and mark the closure of today's lecture. Dr. Ritesh Vishwa.
Thank you, Sandipan. And yes, it was indeed a very nice lecture by Dr. Ritesh Banerjee. I mean, it's always pleasure to listen to him, and we are fortunate to have him with us today also. Thank you, Dr. Banerjee, for your wonderful presentation. Thank you,
sir. Thank you so much.
Prasan sir, for being present with us as a special guest for today's session. Thank you, Sandipan sir, for conducting the entire session beautifully. Thank you all the students from all different colleges, right from Kalimpong College, Parimal Mitra College, and in-house students from the Department of Zoology and Botany, who are patiently listening to the speaker. Although I was expecting more interactive session as the speaker was, but anyways, I hope you have learned a little bit from whatever he has explained to you people. And trust me, these topics are very much relevant, very much pertinent, and these topic will, come to you again and again during the course of your higher education. It will very much needed. Thank you very much, everyone. It was a wonderful session. Thank you for being with us for today's session. Good night, everyone.